Hello, my name is Niall McKay and welcome to Living with PSC, a podcast about the rare disease of primary sclerosing cholangitis. Now, PSC causes the bile ducts inside and outside the liver to become scarred, narrowed and eventually blocked, and leads to liver cirrhosis. In this podcast, we aim to dispel some of the myths about the disease. Well, welcome Dr. Ruth-Anne Pai, who is the new Director of Research Strategy with PSC Partners. Ruth-Anne, it's great to have you on the podcast today. Can you tell us a little bit about what your job entails?
Sure, Niall. It's great to be here with you too. So as the Director of Research Strategy, my goal is to support all of our research programs at PSC Partners. So this includes our research grant program, but also this new strategy that we have to identify the most important areas of research and really initiate and facilitate and drive those projects forward. So this includes aspects like our patient-reported outcome measure project, as well as our natural history project.
Would you like to explain what patient-reported outcome and natural history are?
Absolutely. So let's start with the patient-reported outcome measures. So we call these PROMs for short. And PROMs are short surveys that we can use to capture a PSC patient's experience with their symptoms and how those symptoms affect their function. And it's very important to capture these because unlike other measures that we use in clinical research and clinical trials, these PROMs come directly from PSC patients. And so these are important to the FDA, for example, in clinical trials to understand how a drug that we're testing can impact the quality of life and the symptoms that PSCers experience.
And that's because, say, with another disease, whether it's a sort of a progressive liver disease, for example, you can tell as people are, either progressing or regressing with their illness via, clinical studies or blood tests and stuff like that. But of course, with PSC patients, that's much more difficult, isn't it?
Yeah, exactly. Those tests don't tell the whole story. And so we hope to develop a patient-reported outcome measure, or a PROM, that really captures all of the most important aspects of PSC symptoms that are defined and prioritized by the PSC patient community. And this will be used then for clinical trials. When a PSC patient joins a trial, we hope that this PROM will be given in order to capture those symptoms and how they change over time. So the second project is our natural history project, which at the moment we're really focused on identifying two key pieces that are important for clinical trials for PSC. And we can certainly talk about all of the challenges that we experience in developing clinical trials for PSC. But the two pieces that are really important here are what we call endpoints, which is within a clinical trial, when we are given a drug, what aspects of PSC do we expect to change? And right now, we have two problems. One is that there is a placebo arm for the majority of these PSC trials, which means that PSC patients who enroll may not be given drug at all. And these trials then also go on for years at a time just to be able to measure these hard clinical endpoints like progression to cirrhosis or the need for liver transplant. So our natural history study aims to identify first surrogate endpoints for this long-term progression of PSC. So rather than waiting 2, 3, or 4 years to progress to cirrhosis or need for liver transplant, we're hoping to identify blood tests that we can use as what we call surrogate endpoints that will measure something that is likely to predict progression, instead of having to wait all of that time. So the surrogate endpoints are the first part of the natural history project. We're also hoping to develop through this data collection what's called an external control arm. And the goal or the aim of an external control arm is to replace the placebo group in a clinical trial. So rather than PSCers needing to be on placebo for years to understand whether a drug is beneficial for the treatment group, we can use surrogate endpoints and external control arms developed through this natural history project to shorten these clinical trials and to ensure that all patients can receive drugs.
There's been very little in the way of drug development for PSC, isn't that correct?
Absolutely. And those two challenges, having the patient reported outcome measure, having surrogate endpoints, and having an external control arm, we hope will accelerate and improve the rate of success for these clinical trials.
What has changed the game for PSC Partners? Would you like to talk about the grants that have come in recently?
So we have been funding research grants for years now, and we funded more than $5 million US dollars worth of grants through our grant program. And this year we've received arguably some of the best proposals that we've ever seen. And when we think about funding research grants for PSC, we think about the most important research questions and also research projects that are likely to lead to new therapeutic strategies or drugs that we can try for PSC. And we can also think about other research projects like those that are developing new models that the field can use or generating new questions or hypotheses as also being important. So this year, we at PSC Partners funded 7 grants, and PSC Partners Canada funded 2 grants that are aiming to further understand the biology of PSC and the treatments that we can consider.
Could we just talk a little bit about this Chan Zuckerberg grant and what that means?
PSC Partners was super fortunate to be awarded the Rare as One grant in their initial cohort. The Chan Zuckerberg Initiative, or CZI, has an entire arm of their organization called Rare as One, which aims to focus on rare diseases and developing and supporting rare disease organizations like PSC Partners as we strive to find FDA approved or new treatments for our diseases. CZI has a mission that there are about 7,000 rare diseases, and these diseases affect more than 400 million people globally. And for the vast majority of them, they aren't well understood. And like PSC, fewer than 5% of these rare diseases have approved treatments. So we are fortunate in that we are a part of this cohort of 30 organizations who are supported with a grant from the Chan Zuckerberg Initiative to grow our research programs and to develop a collaborative international research network.
Haven't you guys kind of come up with a strategy where you've identified the areas that really need to be researched and sort of encourage people to apply for grants in those areas?
So this is called the Collaborative Research Network Model, and it was developed by David Fagenbaum at the Castleman Disease Collaborative Network. They published a paper on this model a few years ago, and it includes 8 steps that PSC Partners is now on our way following in response to our CZI Rare as One grant. And the goal of this network is to support our community and to identify the most important research questions. And it's a shift in how we fund research. We're no longer, sitting on our heels hoping that the right researcher will apply to us, will submit a grant. We'll continue with our grant program. We'll continue to support these really high impact research projects. But now we as a community are prioritizing with the patient voice the most important questions. So the 8-step process begins with community building where, Niall, we're really in a good place. We have an amazing PSC community. And through that, we can crowdsource and prioritize research questions, identify the top researchers to study these questions. We can fundraise, procure samples, and get that work done with the best researchers. And really importantly, we have this amazing community, and we need to share this knowledge back with everyone to continue this iterative process. So this really active push that we're developing aims to prioritize research questions that are important to our PSC patient and caregiver community. And it really did come about through the support from CZI and the Rare as One grant.
Would you like to tell us those areas that you've identified as the most important areas?
Absolutely. So at this point, we spent a lot of time at PSC Partners thinking about what the important areas are. And we've also been collecting questions throughout the roadmap series from our community as well. And it's going to be still a long process to condense and prioritize those most important questions. But across, what areas are most important to our community, a few that we've identified are within the clinical research space. Like I mentioned before with our natural history project, identifying surrogate markers and developing a patient-reported outcome measure. This really came out of this understanding of the questions and the needs that the PSC community has. We also see a lot of need to focus on patient care and natural history to understand time to diagnosis and the standard of care for PSC. Within the basic science side of things, there's really a need to understand the relationship between the gut-liver axis and the role that IBD plays in PSC. So that's really an area that we hope to focus on with Dr. Josh Korzenik as well. And another piece is thinking about how PSC Partners can support all of research through providing biospecimens or samples from the community to allow these research projects to take place too.
For example, one of the areas that has emerged is this idea of what's popularly known as the microbiome, the conversation that goes on between intestine and the liver.
Yes, that's one of the really key areas to investigate.
So would you like to tell us a little bit about the grants that you've funded this year?
One of the grants that we funded this year aims to investigate bile acid synthesis and the mechanisms that our body has to regulate the production of these bile acids. And Dr. Wei's grant, which is titled The Role of E3 Ligase HRD1 in Bile Acid Synthesis in PSC, really hopes to identify new drug targets to limit bile acid synthesis in PSC. And we felt this could be a really interesting and novel treatment approach for PSC.
The bile that's produced in the liver is often creates problems for PSC patients, doesn't it? Because it erodes the bile duct and then backs up into the liver and creates cirrhosis. So the idea here is to be able to limit the amount of bile that does that or that creates those problems.
Exactly.
We all know that it's the repeated infections and then the continual scarring caused by bile or caused by infected bile that causes us problems.
Yeah, that's a really major component of it. And then just a second piece is the component of the bile. So sometimes there are more toxic bile acids within the bile in PSC too, and that's thought to be a problem. But yes, this grant really focuses on limiting the production of bile, which can lead to erosion and damage to the bile ducts in PSC. The next one is around cholangiocarcinoma.
Cancer of the bile ducts generally, isn't it?
It is, yes. So Dr. Hove, we are funding his grant called Targeting GPR35 to treat cholangiocarcinoma. And what's really interesting here is that Dr. Hove's group was actually the first to understand what role this protein, GPR35, plays and to understand what this protein really is. And they're planning to perform what's called a drug screen. They're using cell lines and testing promising drug compounds. And they're also using this very interesting experimental model called organoids, as well as mouse models, to test those drugs and determine whether they can prevent the development and progression of cholangiocarcinoma in these organoids and mouse models.
What are organoids? Are they artificially grown organs?
They are. It's a really interesting research area. They've been in development and of interest to many research fields for about a decade now. And the idea is that if we can take human tissue and grow a small organ in a dish, we can then investigate in a 3D structure and manipulate that tissue with drug.
Obviously, the alternative would be a lot more difficult. You'd have to be opening me up to have a look at what's going on.
Right. We either have to open you up or use mice, which we know don't always really reflect what we see within a human PSCer. So the third grant that I'd like to discuss is from Dr. O'Hara, and this one's titled Do Bet Proteins Influence the cholangiocyte response to injury, and are there therapeutic targets for PSC? So the key terms here are BET proteins and cholangiocytes. Cholangiocytes are liver cells, so we can just think of them that way. And BET proteins are really interesting. They're able to read and interpret the 3D structure of our DNA. So we often think of our DNA as just a long line and it gets read to produce RNA and then protein. But in actuality, our DNA is structured and organized and folded. And that process is really important for how our cells function. And Dr. O'Hara thinks that there's some problem with BET proteins and how the DNA is folding in liver cells. So they're hoping to identify drugs that can inhibit these BET proteins to slow the progression of PSC.
We talked to Dr. O'Hara in the last podcast, and he was fascinating because he was also talking about how the cell linings in the tube and the stasis, whether cells died or not, or whether the dead cells were being cleared or not, was causing part of the problem with the scarring that was happening in our ducts, in our bile ducts, essentially. And the fourth area.
Yeah, so the fourth area is a grant that PSC Partners Canada is funding this year, and that's Dr. Richard Green's proposal, which is titled Unfolded Protein Response-Based Therapies of PSC. And Dr. Green's group has previously shown that this process called the unfolded protein response is important in the progression of PSC and in causing liver injury. And this is a study to determine whether a drug that targets XBP1 in the liver could reduce liver injury and progression. And they're using this really interesting method called nanoparticle delivery, where you encapsulate, you create this system of tiny particles that cover the drug and target it for the liver to ensure that we're getting the drug to the right place at the right time, which I think is just really novel and innovative. So I'm very excited to see how that work goes.
Yeah, because I know with a lot of drugs, isn't it? I mean, particularly with cancer drugs, the big job is to get the drugs to the cancer or to the specific area, because often they do a lot of damage on the way down, if you like.
Yeah, absolutely. And that's really where these nanoparticles first started, was understanding how we can deliver drugs to the tumor and cancer. And it's exciting to see now these methods being applied to PSC too. And there's actually one other grant based in that same area of understanding what changes in our gut and how we could potentially target those changes with therapeutics like drugs to slow the progression of PSC. And that grant is from a young investigator, Dr. Elias, called Unbiased Tissue Metabolomic Profiling of PSC IBD. And their goal is to characterize all of the compounds in the colon to identify novel pathways altered in PSC patients with ulcerative colitis. And this could lead to the identification of multiple targets to really understand and alter the changes in the microbiome and the metabolites that we're seeing produced in PSC.
I mean, there's certain bugs that have been identified, or bacteria that's been identified, and the toxins that they produce, if you like, maybe, or the balance of the toxins of which they produce may be contributing to PSC.
We often refer to those as metabolites. The metabolites that are produced in our gut many of them are supported or produced from the microbiome or with support from the microbiome. And those include our bile acids, but also these things called short chain fatty acids and tryptophan metabolites. And so understanding the role of these metabolites in PSC and whether we can alter their production and their secretion into the liver will be really important for PSC too. So there are 2 grants focused on waitlist prioritization. And then there's a grant from Dr. Carlson around bile duct on a chip.
Yeah, should we talk about Dr. Carlson and then the other 2?
Yeah, it sounds great. So Dr. Carlson was awarded a grant this year titled PSC Studies Using a Bile Duct on a Chip. This work aims to develop and validate an experimental and also a preclinical model for PSC called organ on a chip. And with this tool on hand and available to collaborators, Dr. Carlson's team will investigate the interactions between liver cells and immune cells with bile. So 3 really important aspects of PSC: the immune system, the liver cells, and our bile acids. And once they've developed this system, they'll introduce immunomodulatory drugs and determine whether they can inhibit the processes that we see in PSC while we have this bile duct on a chip.
And sorry, what is a bile duct on a chip?
What Dr. Carlson's group will do is they'll take a small amount of cells and place them onto a chip, and they will grow and develop into structures that include bile ducts. And his model will also include immune cells, and he'll introduce bile as well so that he can study the interactions between these components and determine whether drugs are able to inhibit these inflammatory processes that we see in PSC.
So it's a bit like an organoid, but it's almost creating a kind of a virtual environment that they can investigate.
Similar to our question of how do we study the human liver without disrupting our own human bodies, we can develop these organ-like structures like organoids and bile duct on a chip to study liver cells and all of their factors that they interact with in the lab.
It's 22nd century, let alone 21st century.
I totally agree. When I first started in a research lab about 10 years ago, I was reading papers about these structures, and it just seemed impossible that we could create an organ with just a few cells. But here we are, and it's just fantastic to see all of these approaches applied to PSC.
Finally, to wrap up, what were the other 2 areas?
Final 2 grants to discuss today are both focused on understanding waitlist outcomes and liver transplantation in PSC. There is a grant from Dr. Korkompetis titled Update on PSC Liver Waitlist and Post-Transplant Outcomes. And this work seeks to further our understanding of waitlist outcomes and post-transplant outcomes in order to identify and quantify inequities in PSC. And since we now see within the transplant world this movement towards continuous distribution of organs in the coming years, this work would support PSC patients by providing concrete data to support more positive post-transplant outcomes in PSC.
This is a completely unique disease in that way, isn't it? Insofar as that the difference between somebody doing pretty well and doing pretty badly can be weeks, as opposed to with other types of liver disease where the progression would be over perhaps years.
Absolutely. And that's a really good introduction to our final grant, which aims to support applications for exception points for PSC patients on the waitlist in Canada. And this group is led by Dr. Bott, and their grant is titled Closing the Gap, Machine Learning Evaluation of Liver Transplant Waitlist Prioritizations for Patients with PSC. And this is another grant that PSC Partners Canada will be funding. It really aims to support additional exception points for PSC patients on the waitlist in Canada. And we hope that this will have a really strong impact and support our PSC community up there in Canada.
I mean, that's all over because the MELD system doesn't really favor, in fact, it sort of discriminates against people with PSC, doesn't it?
It does. And this is one of those areas, as we were discussing earlier, of unmet need and patient prioritization, where PSC Partners felt very strongly that these 2 areas of waitlist outcomes, of exception points, are really critical to further characterize and understand both in the US and in Canada. And this type of work will hope to support internationally as well.
You're working on trying to delay the disease, trying to help the outcome of people who are on lists, and also trying to help people manage symptoms, develop work that manages symptoms. So I really admire the work that you're doing. It's great. Is there anything you'd like to add to wrap up?
One aspect of PSC that we don't quite understand yet, even though a lot of people have a lot of researchers have investigated and asked these questions is what the true prevalence and incidence of PSC is across the world. So prevalence refers to the proportion of people who have PSC during a particular time. So we could say in 2020, about 10 of 100,000 people in a specific population were living with PSC. And incidence refers to the proportion or rate of people who develop PSC during a particular time period. So for that, we typically say about 1 of 100,000 people will be diagnosed with PSC. But there's a really wide range here. And where the research has developed in recent years is that in just this year, in 2021, Dr. Pollock Trivedi led this really large meta-analysis to understand how prevalent PSC is and what the incidence is. And I'm happy to speak to that research if you'd like.
Yes, please. Yeah.
Sure. So his team found 16 studies documenting incidence and 14 documenting prevalence. The majority of these studies are from Northern Europe and North America, which is a slight, bias in the field, and that we're really asking these questions just in 2 major regions. The highest reported incidence of PSC was about 1.5 people per 100,000 living in Finland and also in the US and Minnesota. It was found to be the lowest in Italy at only 0.1 people per 100,000. The prevalence of PSC also ranged from 31.7 people in Finland and 23.99 in Minnesota to just 1.33 in Singapore and really almost 0 in Alaska. So these studies suggest this wide range in incidence and prevalence. And there are many reasons why we might see that. And we can also discuss some of those limitations and explanations too, if you'd like.
Well, I presume that one of the things that influences it is that the Northern Hemisphere might have a better developed medical system than a lot of the Southern Hemisphere. Would that be correct? Or?
Absolutely. So in order for us to estimate incidence and prevalence, we need to be capturing individuals. We need to be understanding in their medical records that a physician has diagnosed them with PSC. And we know that there are a number of reasons why someone might not be diagnosed with PSC, even if they do have this disease. But another challenge is that when we're only looking at the largest centers or transplant centers, we're overestimating or not capturing the full population. So Dr. Cyril Poncione performed a really amazing study in the Netherlands where he looked at time from diagnosis to transplantation, either in transplant centers in the Netherlands or across the entire country. And he found that when you look at the median time of survival until liver transplant across the entire cohort, it was around 21.3 years. But if you look just at transplant centers, he saw 13.2 years. So it really suggests that you do need to reach out to communities to understand who has PSC and who could use additional support and clinical care.
So there were more transplants or a shorter time to transplant around transplant centres than there was in other areas. Is that the supposition?
That's it, exactly. Yeah, and there are some potential reasons for that. We know that healthcare inequity exists globally. For example, we recognize that multiple racial and ethnic groups are disadvantaged by the American healthcare system, and really very little work has been done to identify and understand the extent of healthcare inequity among the PSC patient community in the U.S. There is a large Latinx population with PSC that's either misdiagnosed or underdiagnosed, because when clinicians will see their Latinx patients, they have been taught through their medical training that they should consider NASH first. And it's true that there is a large population of NASH patients who are of who identify as Latinx, but we also need to consider in that differential diagnosis that this patient should also be considered for a PSC diagnosis as well.
I see. Yeah. So really, with people who are presenting with liver disease, if they, because I think they said I think in Norway, speaking to them, they said that everybody with IBD gets tested for PSC because it's prevalent.
Right. And that's a really important area. It's something that we're discussing with our clinician collaborators is, do we need to consider screening internationally, globally for PSC amongst all patients with IBD? It's something that we're discussing with Dr. Josh Korzenik, and we hope to support research and natural history studies that aim to understand the impact of screening, both in blood, but also through colonoscopy for PSC for all IBD patients.
I see. Are there other markers for PSC other than, say, liver biopsy or imaging, which seem to be most popular at the moment? Problem shows up with the blood, and then I don't think people are taking biopsies as much of the liver anymore, but I think they do a lot of it through imaging, don't they?
They do. They do a lot of it through either imaging or through the colonoscopy, looking at the specific, what they call phenotype, or how the IBD looks. And if there's this right-sided dominance to the IBD, that could suggest additional screening for PSC. But where I really see the field moving in the future is having a more nuanced understanding of not just which blood tests, but the ratios or the differences that we would consider to suggest additional screening for PSC. We'll identify new biomarkers in the blood. We can also look at the microbiome and how the microbiome is different between PSC, IBD, and IBD alone. And with increased screening and genetic testing of fecal samples or bile samples or bile acids in fecal samples, we may also be able to identify those with IBD who are at risk of developing PSC and further screen them, because it really is important to capture PSC early in the disease progression so that we can best manage and treat and prevent progression of PSC.
Well, Ruth-Anne, this has been a great conversation, and I'm so happy that we have you on our team. So thank you very much.
Thank you so much, Niall. It's been such a pleasure to have this conversation with you.
Well, I'm afraid that's all the time we have for this week. For more information about PSC and for a list of people and topics we'll be covering in the near future, please go to www.pscpartners.org. That's www.pscpartners.org. PSC Partners is a non-profit organisation dedicated to providing education and support to primary sclerosing cholangitis patients. There is information and resources on the website. There is a conference once a year. There is a private Facebook group that you can also join. Also, please join our patient registry. This is an anonymous database that provides research support to the medical community who are researching this disease. Thank you very much for listening.